Hello and welcome to Module 8, where this module will be considering Chapter 11, entitled Gene Expression and Epigenetics. And I'll mention up front, the textbook does a less than stellar job of presenting gene expression to you in Chapter 11. So with that in mind, most of the material in this lecture will be material you'll learn simply by watching this lecture video, not as a result of spending considerable time in the textbook. This lecture will serve as an introduction to some of the principles and introductory information surrounding gene regulation. And by providing you with such an introduction, the takeaway from this, the takeaway should be the answer to the question, why must we regulate gene expression? Why is gene expression necessary? Why is it important? In this chapter, we'll open with a discussion of gene expression, what it is, why does it matter? Ultimately, we'll take the material, this module, a bit further than the glimpse we saw last chapter. And in doing so, we'll look at some of the examples to help us see a very big, broad picture. We'll consider animal cloning. We'll look at cortisol as an environmental factor impacting the regulation of gene expression. We'll look at and discuss the lac operon, a group of genes in Escherichia coli or E. coli that those genes regulate the way by which that bacteria pulls in nutrients, whether it's glucose or lactose. Then we're going to dig right into the various ways by which gene expression can be regulated. We'll consider transcription factors, or DNA binding proteins, sometimes they're called. We'll look at the function of enhancers and silencers. We'll be looking at the promoter region. Then we'll consider our chromatins specifically by looking at histones and how modifying histones can lead to regulation of gene expression, as well as we're going to look at DNA methylation itself. We'll consider a concept called alternative splicing. We'll examine the need for mRNA degradation, and finally, we'll look at the degradation of protein, a post-translational activity involved in the regulation of gene expression. Ultimately, this module just skims the surface of a handful of concepts available for regulation of gene expression. There are countless more ways that I won't discuss in this lecture. Rather, just know that this lecture is to serve the purpose of giving you some exposure, ensuring you have some general familiarity with the regulation of gene expression. Our genome contains all of the information, specifically all the DNA sequences we'll ever need to manufacture all of the proteins and all of the RNA molecules that we'll ever need for our entire lives. And that's quite a statement, because when you think about it, we need different amounts of different proteins synthesized at different stages of our lives and in different tissues. For instance, our epithelial cells, it's the ones covering our arms and our legs, our face, those particular epithelial cells need keratin. But our muscle cells need contractile proteins in order to contract. Specifically, they need proteins actin and myosin. Why don't skin cells make actin and myosin? They have the genes stored in their DNA to do so. And why don't muscle cells make keratin? They also have the genes stored in their DNA in muscle cells to do so. We learned in Chapter 10, gene expression is a process by which information in a gene is used to make some kind of gene product. And from here forward, we're talking about protein. And we followed that pathway, being guided by the central dogma of molecular biology right here, which states that DNA goes to RNA. Specifically, we learned that that was mRNA. mRNA goes to some kind of a gene product, commonly a protein. The process of transcription happens in the nucleus. The process of translation happens out here in the cytoplasm. We saw how a gamut of enzymes and other proteins helped read DNA, and then how ribosomes, with the help of tRNAs, help make polypeptide chains from that mRNA transcript that we made. We have a foundation in gene expression, but what does it mean to regulate gene expression, and why is regulation needed? Gene regulation encompasses the mechanisms that determine the types of cells in which a given gene needs to be transcribed. When, for how long, those are questions that our body answers. Often the mRNA transcript is going to be translated in the cytoplasm. The duration of time before that given mRNA molecule or transcript is degraded really depends on how much of that particular protein we may need. And by stating this, the when, the where, the how long surrounding transcription and translation, I hope you can begin to see how important it is that the regulation of gene expression is critical. There are certain times our body needs certain proteins, certain times it doesn't. In fact, our cells are constantly monitoring and responding to changes in our environment. And our environment, it's our bodies. It can be outside our bodies. From the time we're a single-celled fertilized egg up through old age, we need to be monitoring how we're making proteins, when we're making proteins, and why. And this isn't specific for us either. Even the simplest bacteria have mechanisms by which they switch genes on and off depending on environmental cues, and we'll be looking at one of those examples. As we begin to study this chapter, I want to give you an example of why gene expression is so important. In this slide, we see 3 different cells. And if you've taken A and P from me, you know what these are. But we have a neuron here. We have a lymphocyte, which is one of our 5 types of white blood cells. This is a white blood cell that goes on to produce B cells or T cells, which are involved with our adaptive immune system. And this is a typical group of epithelial cells. Specifically, this is skin. And up here we have these dead skin cells. Remember, we have an epidermal layer that ends up being 4 or 5 layers thick. For the rest of this lecture, I want to be talking about this lower layer right here, that living layer called the stratum basale. So look at these 3 cell types, a neuron, a lymphocyte, and a skin cell. All 3 cells contain the very same genome. There are about 20,300 and some protein encoding genes in each one of these cells, all with the same protein making potential or capability. That is, each cell type has the same instruction manual for making the same proteins. And all that distinguishes them from one another is the proteins they choose to make. So take a moment and think about that. 3 cells from 3 very different body systems, one from the nervous system, another from the immune system, a third from our integumentary system. And all that really differentiates them are the proteins they make with each of these cells or within each of these cells. And those proteins, the ones they choose to make, that is the proteins they're regulated to make in each of these 3 cell types. They go on to contribute to the unique cell shape or morphology that we see, as well as contribute to the hugely different cell function each of these cells have. I think we can all agree these 3 cell types have entirely different functions. And this goes beyond the neuron, the lymphocyte, and the skin cell. Let's consider other cells in our body. Think about the individual function of our liver cells in making bile and filtering blood to take out nutrients from the digestive system and screening for toxins. Think about the various cells of the kidneys as they play their individual roles in making filtrate as well as then going on to make urine. We can consider our pancreatic cells and their endocrine and exocrine functions. The cells lining our small intestine, some are for absorption, some are for secretion of mucus, some help with movement. The list is endless. Suffice it to say, cells are morphologically, meaning they look different, and physiologically, meaning they do different things, distinct. Cells are morphologically and physiologically distinct. All the cell types I've mentioned here have the exact same genomes. And even so, we can say the vast differences between these cell types, it's due to the differences in gene expression. Now, as I've mentioned, in our cells contain all of the information we need for our entire body. Each and every one of our cells contains the same genome with the same instruction manuals for the same gene products. It's simply a matter of what our cells choose to read in the instruction manual that differentiate cells. And to illustrate this point, I want to talk to you for a moment about cloning. The idea of animal cloning goes back many, many decades to the 50s at the very minimum. But I'm actually going to tell you a little story of an event that unfolded in early 1997. Or at least it unfolded to the public in 1997, specifically February 22nd of 1997. Well, I was in my 2nd term of my very first undergraduate genetics course. I was a junior in college. I remember where I was seated in the lecture hall. It was a big lecture hall. And scientists at the Roslin Institute in Scotland announced that day that they'd cloned their very first mammal from an adult cell in the process called somatic cell nuclear transfer. Dolly the lamb had been born 7 months before that, and they were announcing that this lamb had been born. So it was born on July 5th of 1996, and then February 22nd, 1997, it was announced that the lamb had been born. Edinburgh, Scotland, and Dolly's birth proved that specialized cells could be used to create an exact copy of the animal they came from, or at least almost an exact copy, because you should catch me there. They didn't factor in mitochondrial DNA from the egg donor sheep. And what did scientists do exactly? So, embryologist Dr. Ian Wilmot and his team took a somatic cell, in this case a single mammary gland cell of a mature 6-year-old female sheep, a ewe, a differentiated cell that only expressed a subset of its genome, specifically the proteins needed for milk production. And then scientists took another cell. They took an egg cell from another ewe, so another female sheep, and removed the pronucleus from the egg cell in a process called enucleation. So we have an empty oocyte Wilmot then took the nucleus from the mammary gland cell and set it next to the enucleated egg cell. And with some very special lab techniques using electrical impulses and all sorts of other chemicals, the mammary gland cell nucleus and the enucleated oocyte ultimately fused together and grew into a blastocyst in the laboratory. And this is actually an expanding or hatching blastocyst. It's not the blastocyst from the experiment. At approximately day 6 following the fusion of the two cell types, when the growing embryo has that nice pretty hollow structure you see here, they transfer the blastocyst into the uterus of a surrogate sheep. So we have 3 sheep contributing to this experiment. We have the sheep by which the udder cell has been taken. We have a sheep by which an oocyte has been taken. And now we have a surrogate sheep by which a blastocyst will be transferred into. So why is all of this important? First, it was the first mammal that was cloned, and that's exciting. And it was very exciting for me that particular day, and even that later that week, as we sat in our genetics hall and listened to our professor go on about the technology that was involved and his fascination in that experimental journey. But important to this class, and the reason I'm telling you this story, the differentiated mammary cell held within it all of the capabilities to give rise to a lamb. A differentiated mammary gland cell was used to create a new lamb. And how did that work? How could the genetic material from a mammary gland cell create life in the form of a new lamb? Well, remember, the genome in the mammary gland cell contained not only the subset of the genes for being an udder cell to produce milk, but it also contained the entire genome for all of the cells in the sheep's body. And I do want to mention some numbers here because I'm making this sound very simple, but this was 10 years of research for Dr. Wilmot. In this particular experiment, it included 277 somatic cells that were fused with 277 anucleated gametes. So those eggs that were devoid of genetic material with the exception of mitochondrial DNA. From there, 29 eggs survived with viability, being transferred into the uterus of various sheep. And of those 29 transfers, those blastocyst transfers, only 1 lamb was born, and that was Dolly. Once you accept the premise that every single cell contains the entire genome, the question becomes how? How does a single cell choose which genes to express and which to silence? How does a single cell promote the expression of some genes while inhibiting the same in others? Cells don't have the ability to consciously choose what genes to express and what to silence. So we need to take a step back and talk about something called housekeeping genes. Many genes in our genome are housekeeping genes, genes that encode for certain proteins, such as those that are important in metabolic activities, making ATP, other cellular components, things that must be expressed constitutively. That is, they need to be expressed at low levels in cells at all times. Such genes might code for proteins involved in cell respiration, because we all need cells to produce ATP. And as a result, they must have the machinery to support cell respiration. Or maybe they're genes encoding for proteins at the cytoskeleton, that internal scaffolding of a cell. All cells need structural support, that scaffolding, and so all cells express those genes involved in making the cytoskeleton. Another example, all cells need histones. Every single cell needs a mechanism to package up the 6 feet of DNA found within the nucleus. And so genes associated with histone production are also housekeeping genes. And these are just 3 examples of housekeeping genes, making housekeeping proteins, the gene products all our cells need to make all the time at low levels. But in addition to housekeeping genes, all cells rely upon individual cells and tissues have their own unique set of gene requirements for cell function. We call these cell-specific or tissue-specific genes. And the expression of these genes gives each particular cell its own particular function and identity. They're the special genes that encode for proteins that allow a neuron to be a neuron, a lymphocyte to be a lymphocyte, or an epithelial cell to be an epithelial cell. And just to clarify, I'm not saying that neurons have their own special set of genes that lymphocytes or skin cells don't have. All that I'm saying is that all of our cells of our body have those genes, but it's only in neurons that the special genes are expressed. to make a neuron. These genes are otherwise turned off or silenced in the lymphocytes or skin cells, etc. Okay, so I think I've emphasized the point, haven't I? Ultimately, we have housekeeping genes all our cells need to express all the time. We call this constitutive, being expressed all the time and in low amounts. And then we have the more unique genes, cell-specific genes, those that need to be regulated by either being turned on or turned off and expressed only in the cells that need their gene products at a given time. Most cells can alter their gene expression in response to cues or signals from the external environment. And in saying gene expression is altered by cues or signals from the external environment, it's important to keep in mind for any given cell in our body, the term external environment may simply mean some place outside that given cell, even within our body. We could mean picking the cues up from the extracellular space surrounding the cell in question. We could mean signals that are coming from nearby cells or even cells far away in other parts of the body. We could mean hormones or other biomolecules, be them in the bloodstream or other extracellular fluids. All of these things serve as external cues, even when still inside the body. And yet, don't forget that the external environment can certainly mean outside the body too. Now, the cues. What about the cues? A single cue in the external environment can have contrasting effects. The cue may turn some genes on and turn others off simultaneously. And all that I'm meaning here, a single cue can affect one cell type in one way and one cell type in another way. So here we have an example of cortisol, and this is a good very general example. So cortisol is a long-term stress hormone. As compared to adrenaline, that's short-term stress hormone that triggers the sympathetic or fight or flight branch of our autonomic nervous system. Cortisol is long-term stress. It has a number of effects on the body. Increased concentrations of cortisol will lend to an increase in protein breakdown. We call that protein catabolism. It lends to an increase in gluconeogenesis, or the making of glucose from non-glucose sources. It decreases glucose uptake by muscle cells and increases our ability to break down dietary lipids. Glucose can decrease bone formation as a result of impaired intestinal reabsorption of calcium, as well as the increase in the number of osteoclasts in bone. And if you've taken A&P from me, remember, osteoclasts are responsible for breaking bone down, osteoblasts are responsible for building bone up. And really, I'm not all that concerned about you knowing these 5 things, but I just want to show you that there are increases in some things here, increased protein breakdown, increase in the making of glucose from non-glucose sources, increase in breakdown of lipids, dietary lipids, but decrease in glucose uptake, decrease in bone formation. These are all unique and different activities. They are an environmental cue associated with higher than normal levels of cortisol in the body. So they're activities that happen in different places. Keep that in mind. And cortisol targets all of these cells, either turning genes on or turning genes off, depending on the cell type. Gene expression can be regulated at any point in the process of gene expression. So we can regulate at any step involved in gene expression. And if we look at the central dogma of molecular biology, which says DNA is transcribed into mRNA, mRNA is translated into an amino acid chain, and then at some point, with some modifications, we end up creating a protein. We can target gene expression at transcription, and this is where we're going to focus most of our time with the rest of this lecture. However, there are various other targets as well. We might target gene expression in the steps involved in post-transcriptional regulation. Remember the process we discussed last module where following RNA polymerase creating that pre-mRNA transcript, we must add a 3' poly tail, a 5' cap. We were going to remove some introns before making a mature mRNA molecule that leaves the nucleus. The remainder of the steps here we won't spend all that much time on, but we can target gene expression after the mature mRNA leaves the nucleus. We can control how quickly mRNA degrades, and I'll talk about that a little bit. Because think about it, we can't have the mRNA molecule or transcript in the cytoplasm being translated indefinitely. A time will come when we no longer need to translate a given mRNA transcript. And there are enzymes that are going to be responsible for breaking that down. We can also control the process of translation, that is with the help of ribosomes and tRNAs, or we can even control protein activity by determining how long a given protein will remain active in a cell before it's targeted for destruction. And I'll show you a bit about that at the very end of this lecture. So I'll pose this question to you today. We're going to spend most of our time looking at gene expression at the level of transcription. So how do we regulate gene expression? We're going to spend most of our time looking at that from the transcriptional level, although I'll briefly provide you with some other common examples. But why focus on transcription? Why will we spend most of our time at the transcription level of gene regulation? In fact, why do cells spend most of their time at the transcription level of gene regulation? It's the most common target for regulating gene expression. Think about it. Think about why. What have we invested here when we're looking at these steps as compared to here? What are we investing here as compared to here? If we're a cell, what kind of energy have we invested in the process of making a protein if we regulate gene expression early on, at least in the case of silencing genes? Or rather, what energy do we invest by making the mature mRNA transcript? As we go step by step through this process here, we're expending ATP by adding a poly A tail, by adding the 5' cap, by removing introns. We further expend energy in various other ways as we move the mRNA transcript from the nucleus to the cytoplasm. And further, when we move on to translation, it takes ATP to join amino acids together when we begin creating that polypeptide strand at the site of the ribosome. And so it goes. Making a gene product, a protein, it's expensive. It costs the cell a lot of energy. And ultimately, if we can control gene expression for the production of a gene product from the very start, if we're a cell and we know we don't need a given protein, we can silence the genes we don't need to express at the very beginning or from the very beginning. And in doing so, we're saving a lot of energy the cell can use for other things. I want to give you another example of environmental control of gene expression. In this case, how the external environment controls gene expression. And in doing so, we're going to go back to 1961. French scientists François Jacob and Jacques Monod were studying dietary habits of Escherichia coli, again, E. coli. So we're talking about a bacteria. And as they were studying these dietary habits, they noticed something really peculiar. Unbeknownst to them, as they were looking at E. coli food requirements, they inadvertently discovered gene regulation at work. Specifically, E. coli prefers glucose as its carbon source because it requires few steps to metabolize and requires little energy to break down. However, if glucose isn't around and lactose is available, then E. coli will make an enzyme called beta-galactosidase to break down lactose. To use lactose, bacteria must express a certain group of genes involved in the uptake and metabolism, something microbiologists have named the lac operon genes. And E. coli only expresses these genes when two conditions are met. Lactose needs to be available, and glucose shouldn't be available. So, how does E. coli detect lactose? And how does the presence of lactose lend to the transcription of genes involved in the lac operon? Well, let's turn to the tablet and draw it out. First, we're going to have a segment of bacterial DNA. So here's our segment of bacterial DNA. And within this segment, we're going to find three regions. We're going to find what's called promoter. We will find something called an operator. And lastly, we're going to find the coding region. So the coding region is going to encode for the enzyme beta-galactosidase. So beta-galactosidase, which is an enzyme that breaks down lactose. And we have one more thing to include here. We're going to have something called a repressor, and I'll draw the repressor out. Now, under normal conditions, in the presence of glucose, we have what's called this repressor here, a protein that's ultimately going to fit right into this operator region. See how these structures are going to line up kind of like a lock and key fit. And that's great. When the repressor is in this form, it's going to join with the operator and serve as a barrier for RNA polymerase. So RNA polymerase might wrap itself around the promoter, but when this repressor is positioned here in this spot, it will prevent, we won't be able to move forward and move downstream. So the repressor, when in position, it is going to prevent RNA polymerase from moving forward, and we won't see gene expression. However, in the presence of lactose, so imagine we have lactose out here. So this is lactose. lactose right here. Lactose is going to bind to the repressor, resulting in a conformational change of the repressor. And as the repressor changes, create some change here. As lactose binds, it's going to change this repressor such that now we have this conformational change. And now we can't insert ourselves into the operator. If we can't insert ourselves into the operator, what that means is our RNA polymerase is going to be able to move forward. And just label RNA polymerase. And so when repressor plus lactose does not bind to operator, then RNA polymerase will transcribe the gene for beta galactosidase. And that will lead to breakdown of lactose. Now, I have one housekeeping item I didn't draw on the tablet, and I should have mentioned it, even though I'm not all that concerned about it in terms of your understanding of gene expression by environmental cues as we look at this lac operon. But it wouldn't be right to leave it out entirely. So referring back to this diagram here, there is a special sequence here that we call the cap region or the cap site. And remember I mentioned earlier that for the lac operon to be expressed, lactose must be available, whereas glucose levels should be low. Well, we talked about lactose and lactose binding to the repressor in order to disassociate operator and repressor. However, I didn't mention what happened with glucose. Something needs to determine low levels of glucose, and we have a chemical called cyclic AMP. I don't really want to get into the nuances of cyclic AMP, but suffice it to say, it communicates with this cap region here when glucose levels are low. And in communicating with this region, CAP makes the promoter region more desirable, a better place for RNA polymerase to bind. Also, and maybe I should have mentioned this, when I talked about lactose binding to this repressor, I should have mentioned that lactose acts as an activator for the repressor. So to recap, we have a promoter region, we have an operator region, we have a coding region. I don't care that you know these three genes, doesn't matter, but you have lac genes within this coding region. We also have a cap region by which cyclic AMP communicates with the cap region in cues for low glucose. And we have an activator, specifically we have lactose, that interacts with the repressor, which binds to the repressor, changing this lock and key fit such that the operator and repressor no longer bind together, thereby ensuring RNA polymerase comes in and can move downstream to transcribe these genes. Okay, so that is the lac operon. And again, this is a bacterial system giving you a good example of how we see gene control or repression, and in this case, we're talking about prokaryotes. Now this stage has been set, let's go ahead and dig into gene regulation at the transcriptional level. Remember from our last module, RNA polymerase begins the process of transcription by binding to a gene's promoter region. Remember we looked at the TATA box, a unique sequence within the promoter region by which RNA polymerase was attracted. And all genes, no matter what the cell type, has additional DNA sequences or nucleotides, usually near the promoter, that help turn genes on or help turn genes off. These DNA sequences that govern the expression of a gene are called regulatory sequences. Regulatory because they regulate, and sequences because they're nothing more than sequences of nucleotides along a strand of DNA. So how do these regulatory sequences work? Well, in fact, regulatory sequences don't actually do anything at all as it pertains to gene expression. Rather, they're merely acting as binding sites, or I've heard them called landing pads, for DNA binding proteins that are called transcription factors. Transcription factors are just a fancy name for a regulatory protein, and it's the transcription factor that does all the work. So I have a couple of images here we're going to look at. This is an illustration of a segment of DNA, and we see transcription factors, this DNA binding protein here in yellow, coming in and binding to a sequence of DNA called an enhancer. The enhancer is a regulatory sequence upstream of a promoter, so here's upstream to the left, And binding of a transcription factor with an enhancer will ultimately promote RNA polymerase binding with the promoter downstream, thereby turning gene expression on. So enhancers are responsible for turning things on. Alternatively, here's another illustration, and we're looking at this little spot right here. It's a segment of DNA where we see transcription factors in orange coming in, these two in orange, binding with a regulatory sequence called a silencer. And a silencer is a regulatory sequence by which binding with a transcription factor ultimately discourages RNA polymerase binding with that promoter that's downstream. And so that's going to be turning a gene off, or gene expression off. Now that I've given you two examples, a transcription factor binding to a regulatory sequence called an enhancer, and another transcription factor binding at a regulatory sequence called a silencer, is it the regulatory sequence that turns the gene on or off, or is it the transcription factor, the binding protein, if you will? Actually, it's neither. It's the combination of these transcription factors binding with the regulatory sequences of the enhancer or silencer that make this work. Without the transcription factor binding to the regulatory sequence, gene expression would not be impacted. And without the regulatory sequence, the transcription factor wouldn't have any way to bind. Thus, it wouldn't recognize the gene or aid in modifying gene expression through the process of transcription. So, because the two act together, we need both the regulatory sequences within the enhancers and silencers as well as the transcription factors themselves to work together to regulate gene expression. Lastly, I'm going to go back to our example of the neurons, the lymphocytes, and skin cells. In this interaction between enhancers and silencers and transcription factors ultimately leading to cell differentiation, how cells become specialized, how neurons become neurons, how lymphocytes become lymphocytes, how skin cells become skin cells. This is how it works. I've gone ahead and just put in a separate slide giving you just a little bit more information about our enhancers and our silencers. Most enhancers are under 20 base pairs in length. Silencers are somewhere between 100 and 300 base pairs in length. So these are it's just a bit more information about our enhancers and silencers. And the last thing I want to point out here, we have what's called cis regulatory sequences. That's a fancy name. That cis simply means these sequences are on the chromosome that we are looking to regulate. If we had trans-regulatory sequences, those would be regulatory sequences in other chromosomes that are regulating the expression of a gene on this chromosome. So cis regulatory sequences just simply represent the regulatory sequences on the same chromosome by which we are trying to undergo gene expression or controlling gene expression. So it's a comment that the textbook mentions, doesn't go into it in a lot of detail, and so I just wanted to clarify that. Okay, histone modifications. Recall from last module, or was that 2 modules ago? We learned our chromosomes consist of DNA complexed with histones, which join together in sets of 8 to form a nucleosome. And then we have some non-histone chromosomal proteins as well. All of this contributes to chromatin. We have tight association of DNA with histones and other chromatin binding proteins that has a somewhat repressive effect on gene expression, as access to the transcription machinery is physically impeded when we have DNA wrapped around these histones that are wrapped into nucleosomes that form these loops that end up forming our chromatin. So access to transcriptional machinery is physically impeded through this process. The structure of chromatin naturally inhibits access to DNA by proteins involved in transcription, amongst other things. Now, we need to revisit the structure of the histone momentarily. Histones are found in sets of 4 proteins, and histones have 2 different parts. They have a globular domain that's made up of our 4 little spools that hang out together. And this is one of them right here. But they also have what are called positively charged tail domains. So something that's positively charged, and we're seeing tail domains here and here and here and here. And those interact with the negatively charged phosphate groups on the backbone of DNA. So DNA is somewhat negatively charged because of those phosphate groups. The tail domains of our histones are positively charged, and that negative and positive interaction is going to hold DNA very close to the histones. The positively charged tail domains are the focus of gene expression when we talk about histone modification. These tails can be modified by adding or removing methyl groups, acetyl groups, phosphate groups. So let's go through each of these three. And this is going to be an illustration for adding acetyl groups. This is an illustration for adding methyl groups. This is an illustration for adding phosphate groups. All right, acetyl groups. One type of histone modification affecting histone structure and ultimately chromatin structure is the process of acetylation, or the addition of an acetyl group, which is CH3CO, and the O has a double bond with it. And that being added to a positively charged tail on a histone protein. With increasing acetylation, with many acetyl groups being added, the positively charged histones slowly lose affinity for negatively charged DNA, ultimately destabilizing chromatin structure and allowing for transcription to take place. And so this illustration here, we're saying transcription off, we add acetyl groups, we're going to encourage transcription. Acetyl groups are added to histone proteins by an enzyme called acetyltransferase, and then there are other enzymes called deacetylases that strip acetyl groups from histones to restore chromatin structure and repress transcription. So we have the addition of an acetyl group is going to encourage transcription because we're loosening things here, which allows the machinery needed to access pieces of DNA or the genes of DNA. We have methyl groups. Methyl groups are CH3 groups. Another type of histone modification is the addition of a methyl group to the tails of these histone proteins. These modifications can bring out activation or repression of transcription depending on what particular amino acid in the histone tail is being methylated. Generally, when CH3 groups are added, methylation spreads from the tail of one histone to the next, to the next, to the next, propagating something we typically see as gene silencing. Generally, the addition of a methyl group is going to turn off gene expression. And so that's why we see that negative sign right here. Enzymes called histone methyltransferases add methyl groups to specific amino acids, usually lysine or arginine of histone proteins. Other enzymes called histone demethylases remove methyl groups from histones. So here we're adding methyl groups, those CH3 groups, to histones directly. We're going to add some more methyl groups to something else in a little bit. So this is an area where that can cause a bit of confusion. What are we actually adding methyl to? In this case, we're adding it to our histones. We have phosphate groups that we might add. Histone phosphorylation can occur on amino acids such as serine, threonine, or tyrosine. So we have a couple of different amino acids by which we can phosphorylate. And similar to acetylation, phosphorylation is generally associated with the activation of transcription. We're positively regulating gene expression. And adding phosphate groups, a negatively charged species, phosphates are negatively charged, to a histone lends to this negative repulsion, or reduces the positive charge of the histone, which provides for chromatin instability, opening these bits of DNA up that are wrapped around. And in this case, we're wrapping around here where if we add a phosphate group, we're loosening some of this. We're loosening that attraction between histone that's positively charged and the phosphate backbone of DNA that's negatively charged. Histone phosphorylation is mediated by protein kinases that add these phosphate groups, and then protein phosphatases remove phosphate groups. Now, these modifications have sometimes been called the histone code because they encode information that affects how genes are expressed. So these are three ways by which we just focus on the histone, and we're focusing on these tails. In our last slide, one of our three examples of histone modifications was the addition of a methyl group to histones to silence gene expression. But another type of chromatin modification, so now we're not looking at the histones, but we're looking at chromatin itself. We can add methyl groups, those CH3 groups, to our DNA bases. Specifically, methyl groups are placed at the site of a cytosine base, found adjacent to a guanine. So we're going to see cytosine and guanines next to each other. And in that situation, we may be able to add methyl groups to that cytosine. These regions of DNA having these cytosine guanine sequences are sometimes called CPG islands. And I've mentioned that here in this slide. CPG islands are commonly found in or near promoter regions. And evidence suggests that DNA methylation of CPGs represses gene expression by inhibiting the ability of transcription factors to bind to DNA. And if this sounds familiar to you, it should. We discussed X chromosome inactivation in a prior module, where we methylated one of our X chromosomes in order to silence it or inactivate it to ensure for females we don't produce too much of a gene product for our body. Because as a female, we have two X chromosomes. We have two opportunities to make any of those proteins that are on that X chromosome. And we need to ensure that doesn't happen. So if this sounds familiar, DNA methylation, this is the way by which we inactivate one of our X chromosomes in every single cell in our body. Moving forward, now we've moved beyond the initiation of transcription, and now we're looking at a pre-mRNA transcript. We have a process called alternate splicing. Now earlier, in an earlier video, we talked about removing introns as part of the post-transcriptional processing experience. We added some poly-A tails, we put on a 5' cap, and we took out introns. Well, in the 1970s, scientists discovered that genes exhibited something called alternative splicing. Recall, mRNA splicing itself is one of those post-transcriptional steps a pre-mRNA transcript takes to develop into that mature mRNA transcript that's then sent from the nucleus to the cytoplasm for translation with a ribosome. But what is alternative RNA splicing? What does that alternative mean? Alternative splicing is a mechanism that allows for the production of different functional mRNA transcripts from the very same pre-mRNA transcript, which ultimately leads to the production of different proteins encoded by the same gene. In this manner, different combinations of introns and exons are removed from the transcript. An example of this, our textbook, I wish that it discussed this, and it doesn't. There's some other textbooks that do. An example of alternative splicing results in tissue-specific production of a protein, and we can focus on a particular gene called the CT-CGRP gene. Sometimes it's called the calc gene. And we see that it comes into play in two ways. In the thyroid gland, calcitonin is produced. And if you take an anatomy and physiology from me, you know exactly what calcitonin does. It's involved with controlling calcium levels in the blood. Specifically, the CT-CGRP mRNA transcript, that's a mouthful, is spliced to produce a mature mRNA that contains the first 4 exons of the mRNA transcript, but exons 5 and 6 are omitted. So if we take this gene and we make the pre-mRNA and it's destined for the thyroid, for a thyroid cell, and we've transcribed this gene, we're going to see removal of some exons 5 and 6. And we usually don't remove exons. What we've learned already to date, we didn't say anything about exons. We only said removal of introns. But here we are removing some exons. In contrast, if we are a neuron, not all neurons, but some neurons, for a neuron, we take the same mRNA transcript. So neuron cells are transcribing this gene as well. And different than what the thyroid cells are doing, they're taking that mRNA transcript and they are removing exon number 4, but leaving exons 5 and 6 in. So now we're removing exon number 4. Of course, all the introns are being removed, but we're removing exon number 4, leaving 5 and 6. And so what we end up seeing are two, because of alternative splicing, we see two different proteins result once we send that mature mRNA transcript out to the cytoplasm to meet up with ribosomes to translate that transcript into protein. Alternative splicing is controlled and acts as a mechanism of gene regulation with the production of different protein products in different cells or at different stages of development. Alternative splicing is now understood to be a common mechanism of gene regulation in eukaryotes, so our bodies as well as others. According to one estimate, 70% of genes in the human body are expressed as multiple proteins through alternative splicing. And to give you some numbers, our human genome is thought to contain about 20,300-ish protein encoding genes. Yet we make upwards of 75 to 100,000 or more different proteins. So we don't have one gene for one protein. In fact, we might take one gene, one pre-mRNA transcript, pull some exons out, different exons for different cells, and in fact, we're making different proteins. Let's consider mRNA degradation. The amount of a protein synthesized depends on the amount of the corresponding mRNA available for translation. The amount of available mRNA, in turn, depends on both the rate of mRNA synthesis, which we can control at the transcriptional level, as well as the rate of mRNA degradation, or breakdown. Some mRNA persists just a few minutes in the cytoplasm for translation, others last for hours, for days, even for months. Cellular RNA, including mRNA, is degraded by something called a ribonuclease, enzymes responsible for the breakdown of RNA. And there are a couple of different pathways for the degradation of mRNA. The first pathway is going to involve focusing on the 3' end, removing those poly A tail pieces through a process we call de-adenylation. This is our Pac-Man pulling off those adenines there. A second pathway involves the removal of the 5' cap, a process we call decapping, followed by nucleotides removed in the 5' to 3' direction. And so here's our 3'. We pulled off those adenines there, but now we're in the 5' direction. We're pulling off that 5' cap, and then we're going to see degradation moving in this direction. Finally, a third pathway, mRNA is cleaved at internal sites with the help of enzymes called endonucleases. Once an mRNA is cleaved, the two parts can be acted upon by exonucleases. So these are just 3 ways by which we can degrade the mRNA transcript such that it isn't being produced for an indefinite period of time. Lastly, now we're at the protein level. Regulatory mechanisms also exist at the post-translational level, especially determining the lifetime of a protein. And there are a variety of ways this can unfold. We might phosphorylate a protein to cause some kind of conformational change, altering the substrate maybe of an enzyme that is supposed to be binding to some substrate. In our last slide of this lecture, I want to focus on the process of protein degradation with the help of a protein called ubiquitin. Ubiquitin is a small, just 76 amino acid long protein that targets a protein for degradation by covalently bonding to it via some lysine side chains. Lysine's an amino acid in a process we call ubiquitination. Subsequently, once ubiquitin attaches, that first ubiquitin, it's going to promote more ubiquitin to come to the area and promote more additions to that. And we see that here. And this is an ATP dependent process. We need ATP for this. And so this process can be repeated to form long polyubiquitin chains extending from the protein. So we see protein here bound to be degraded. We see these ubiquitin. These are just little red flags being waved in the air. They serve as tags that mark the protein for destruction. And so then how does destruction take place? Ubiquitin-marked proteins are ultimately recognized by something called the proteasome, a multi-subunit protein complex with protein-cleaving capabilities. The proteasome unwinds target proteins, removes their ubiquitin tags, and then breaks a protein down into small little chains of peptides. And so that is going to conclude the last of our control of gene expression. Again, this is just a brief overview. In closing, we've discussed the regulation of transcription at various different levels, as well as looked at some forms of translation control involving the degradation of mRNA. We've just looked at degradation of proteins. So this is post-translational. We've touched just a very, 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 very small tip of an iceberg. Careers are made studying individual pathways by which we've covered everything in a matter of, 45 minutes? The takeaway from this lecture should be the understanding that each and every one of our nucleated cells in our body holds our entire genome, and it's only through gene expression or regulation of gene expression that our cells develop their own morphology and their own function. We turn some genes on, we turn some genes off. We do this not just for cell specialization, but sometimes just to save energy. And so this concludes module number 8. If you have any questions, don't hesitate to reach out. And as always, make it a great day.